4 minutes after your heart stops, your brain is still working. Your own gut bacteria are waiting to eat you, and the only thing stopping them is that you are alive. You will go stiff, but not because your muscles tighten, because they can no longer let go. And a century from now, the last trace of you will be a patch of ground where the plants grow better. This is not horror. This is forensic science. Measured, timed, published on real bodies by people who volunteered. 10 stages, minute by minute. Let's begin. Your heart stops. That is the legal line, but nothing inside you has got the message yet. Your brain is holding about 10 seconds of oxygen. It spends all of it. Neurons keep firing. And then in many people, something stranger happens. The dying brain does not fade out. It spikes. Here is the problem. That spike is not noise. It is organized. In 2009, an intensive care physician named Lakshmi Chawla watched the brain monitors of dying patients surge in their final minutes, patient after patient, the same burst. In 2013, Jimo Borjigin at Michigan measured it properly in rats and found the surge was more coordinated than waking life. And in 2022, a team led by Raoul Vicent recorded it in a human entirely by accident. An 87-year-old man died mid-scan, with the electrodes still running. The pattern in his last 30 seconds was the pattern a brain makes when it retrieves memories. Doctors tried to explain it away. Oxygen starvation, random discharge, dying static. But static is not rhythmic. This was rhythmic, the same frequency bands in the same order across species. Nobody can ask the brain what it was doing. So here is what the data allows. For up to a minute after you are declared dead, the organ that is you may still be running. And if Vicent is right about what those waves mean, it may be running your memories. You could not tell anyone. You could not move. The last thing you do might be to watch your own life play, in a room where everyone has already stopped talking to you. Blood has stopped moving. Oxygen delivery is over. Every cell in you runs on a molecule called ATP. You make it with oxygen. Without oxygen, you hold about 4 minutes of stock. Then the machinery stops. And here is where almost everyone gets it wrong. Your cells do not simply switch off. They rupture. Inside every one of your cells sit tiny sacs called lysosomes, packed with digestive enzymes. They are the cell's own demolition kit. While you are alive, you keep them sealed, and that seal costs energy continuously every second for your entire life. When the energy runs out, the sacs leak. The enzymes do the only thing they know how to do. They digest. They digest the cell that was holding them. Pathologists call it autolysis, self-digestion. In 1955, a Belgian biochemist named Christian de Duve discovered those sacs and named them. He won a Nobel Prize for it in 1974. He also understood exactly what he had found. Every cell in your body carries the tool for its own demolition, held back by nothing but a membrane you have to keep paying to maintain. We tried to outrun this. Refrigeration slows it. Freezing slows it further. Cryonics companies race to cool a body within minutes for exactly this reason. Slows, not stops. You are not destroyed by anything from outside. The instructions were already inside you in every cell, in place since before you were born, waiting for the moment you stopped paying. Your heart was a pump. The pump is off. But your blood is still a liquid and gravity never stopped working. Within about half an hour, it begins to sink. Wherever your body is lowest, blood pools and that skin turns a deep purple red. Everywhere else drains pale. Forensic pathologists have a name for each half of this. Pallor mortis, the paleness. Livor mortis, the pooling. Here is why it matters far more than it looks. For the first several hours, the pooling can still move. Turn the body, and the purple migrates to the new low point. But after roughly 8 to 12 hours, the blood cells break down and stain the surrounding tissue in place. Now it is fixed. Now it is a record. This is one of the oldest tools in all of forensic medicine. In 1247, a Chinese official named Song Sai wrote the first known forensic manual, and it already describes lividity and how to read it to tell whether a body had been moved after death. Embalmers try to prevent it by replacing the blood entirely. It works, but only if they reach you fast enough, and it only changes what the record says. it does not stop one being written. Your body becomes a document. It records the position you were left in, how long you stayed there, and whether anybody moved you. And it will keep telling that to anyone who knows how to read it, long after you have stopped being able to correct them. 2 to 4 hours in, your jaw tightens, then your neck, then your shoulders and downward until all of you is rigid. Rigor mortis, everyone has heard of it. Almost everyone has the cause backwards. You do not stiffen because your muscles contract. You stiffen because they cannot stop contracting. Here is how a muscle actually works. Filaments inside it reach across, grab each other, and pull. That is a contraction. But to let go of that grip costs energy, 1 molecule of ATP per bridge every single time. Read that again. Relaxing is the expensive part, not tensing. So when the ATP is gone, the grip stays. Every muscle you have locks into the last position it held, and nothing can release it, because releasing it requires precisely the thing you have run out of. In 1811, a French physician named Pierre-Hubert Niston mapped the order it travels, jaw and face first, then downward through the body. It is still called Niston's law. The whole cycle runs on roughly 12-hour thirds. About 12 hours to spread, 12 to hold, 12 to fade, and it only fades because the enzymes from stage 2 finally chew through the bridges and let go on your behalf. Undertakers know this. Before a viewing, they massage and flex the limbs to break the rigor by force, so the body can be arranged into something the family can look at. For half a day, you hold the exact shape of the last moment you were inside yourself. Whatever you were doing, however you were lying, it sets like plaster. You have been 37 degrees Celsius your entire life. You have never once, not for a moment, been the same temperature as the room. Now you are cooling. Roughly a degree an hour at first, then slower as you close on your surroundings. Algor mortis, the cooling of death. And here is why this stage matters more to the living than any other. That cooling curve is a clock, and it runs backwards. In 1988, a German forensic scientist named Klaus Hentsch published a nomogram, a chart that takes a body's temperature, its weight, and the conditions around it, and hands back the hour of death. It is still in daily use. An investigator arrives, takes one reading, and reads backwards to the moment your heart stopped. The world tried to make that hard. Clothing changes the rate. Wind changes it. Water pulls heat out roughly 25 times faster than air. Body size changes it. A large body stays warm for many extra hours. Hensch's chart accounts for every one of them. And there is one more detail nobody warns you about. Your eyes. If they are left open, the exposed strip of surface dries out and darkens into a brown-black band straight across the white. Pathologists call it tache noire, the black stain. It can appear within hours. You cool, you dry, and the temperature of whatever room you happen to be in becomes one of the last measurable facts about you, a number a stranger will write down to work out when you left. You are carrying roughly 38 trillion bacteria right now. Most of them live in your gut. Alive, they are useful. They break down food you cannot. They manufacture vitamins for you. Alive, they are also contained. Your immune system spends every second of your life holding them at the wall of your intestine. That containment costs energy too. Here is the moment everything turns. When the immune system stops, the wall stops being a wall, and those bacteria find themselves in a warm, wet, nutrient-dense environment with no defenses and no competition anywhere in it. They do not attack you. Nothing so dramatic. They simply spread because there is no longer anything to say no. Within a day or two, they have moved out of the gut and through the tissue into the liver, the heart, the brain. Microbiologists have a name for the community that takes over. The thanatomicrobiome, the microbiome of death. In 2013, Jessica Metcalf and her colleagues showed the succession is so predictable, a different set of species dominating at each stage, that it works as a clock in its own right, accurate to within a few days. Gulna's Javan's team has been mapping the same thing in humans ever since. We tried to stop this too. Antibiotics, sealed caskets, refrigeration. None of it reaches bacteria that are already inside you. The thing that finally consumes you has lived in you, cooperatively, for your entire life. It was never held back by a lock or a wall. It was held back by your effort. Those bacteria are feeding, and feeding produces gas. Methane, hydrogen sulfide, carbon dioxide, ammonia. The gas has nowhere to go, so it accumulates. The abdomen swells first, then the whole body. Skin tightens and takes on a shine. Features distort. This stage is called bloat. 2 compounds arrive with it, and their names were not chosen gently. Putrescine and cadaverine. They are the smell. They are also the reason a body becomes unmistakable to every scavenger for a kilometer in any direction. Here is what makes this more than unpleasant. Human noses are extraordinarily sensitive to exactly those 2 molecules. We detect them at parts per billion, far below the threshold for most other compounds. Biologists think it is an adaptation. An ancestor who could not smell decay ate it and did not stay an ancestor for long. Arpad Vass, a forensic anthropologist at Oak Ridge National Laboratory, has spent decades cataloging the chemistry of this stage. He has identified more than 400 distinct volatile compounds coming off a decomposing human body. Then the pressure wins. Gas and liquefied tissue force their way out through whatever opening is available. Pathologists call it purge. It ends the bloat, and the body collapses back down. For a few days, you are larger than you have ever been in your life. Then you are smaller than you have ever been. And the difference has gone into the air, some of it into the lungs of whoever is standing closest. Active decay. This is the stage where most of your mass leaves. Soft tissue liquefies and drains into the soil beneath you. If insects can reach you, everything accelerates. Blowflies find a body within minutes of death, sometimes before the heart has fully stopped. They lay eggs. Larvae hatch within about a day and feed in a mass so dense it generates its own heat, up to 10 degrees above the surrounding air. Here is the part that genuinely surprised the scientists. You do not simply disappear into the ground. You change it. The nitrogen in your body floods the soil in a single concentrated pulse. Ecologists call the result a cadaver decomposition island, a patch of earth chemically unlike anything around it. At first, that pulse is so strong it is toxic. The vegetation directly beneath a body dies. And then, months later, that same patch turns greener and grows harder than the ground around it. For years, David Carter and Mark Tibbett described and named the effect in 2007. Botanists can now locate forgotten grave sites from the plants growing over them. This is what the body farm exists to measure. In 1981, an anthropologist named William Bass founded a facility in Tennessee where donated human bodies decompose in the open air, under observation, in every condition researchers can arrange. Nearly every number in this video traces back to places like it. You do not leave the world. You are redistributed into it, and the ground keeps a bright green record of exactly where you were lying. A year in ordinary soil, the soft tissue is finished. What is left is skeleton, some cartilage, hair, and whatever the environment decided to keep. And the environment decides nearly everything. A 19th century German forensic physician named Johann Ludwig Kasper published a ratio that still holds today. 1 week in open air does what 2 weeks under water does, and what 8 weeks in the ground does. Air, water, soil. 1, 2, 8. It is called Kasper's dictum, and investigators still reach for it first. But bury a body somewhere cool, wet, and airless, and something else happens entirely. The body fat undergoes a chemical change, hydrolysis, and converts into a waxy, grayish, soap-like substance. Grave wax, adiposer. Adiposer does not rot. It seals. Bodies have been recovered after 100 years with faces still recognizable because of it. And here is the failed rescue. The sealed casket and the concrete burial vault were sold on a promise of protection. What they actually build is an airless container, which is the precise condition that favors anaerobic bacteria and adiposer. Sometimes enough gas pressure accumulates inside to break the seal outright. The funeral industry has a term for that, and it is not a euphemism: exploding casket syndrome. Meanwhile, the box itself gives way. Wood rots. The lid collapses under the weight of soil above it. Every cemetery knows the timeline. The ground over old graves sinks. The container you were sold to keep the world out becomes the reason the process takes a stranger, slower path. Bone feels permanent. It is not. Bone is a composite, a mineral scaffold of calcium phosphate woven through with a protein called collagen. The mineral supplies hardness. The collagen supplies flex. The collagen goes first. Over decades, water and soil chemistry break that protein down and leach it away. What is left behind turns brittle and begins to flake apart. And acidic soil attacks the mineral directly. In acidic ground, a skeleton can vanish completely within a few decades. Change the conditions, and the answer changes entirely. Dry, alkaline, or frozen, and bone lasts tens of thousands of years. Archaeologists call the whole process diagenesis. It means precisely the same thing for you as it does for a mammoth. So the honest answer to how long you last is not a number at all. It is a location. And here is the last fact, the one that outlives every stage in this video. Not a single atom in your body is destroyed by any of this. The carbon, the nitrogen, the calcium, the water, every atom is conserved. It leaves you, and it enters soil, air, roots, and other living things. You were assembled out of atoms that were already ancient, forged in stars, cycled through oceans and rock and other organisms for billions of years before they were briefly, temporarily, arranged into you. You never owned them. You were a pattern those atoms held for a few decades. And every stage in this video is just the pattern letting go. That is the timeline. 60 seconds of a brain that may still be running your memories. 4 minutes to the point where your own cells start digesting themselves. Half an hour for your blood to sink and start keeping records. 3 hours to lock into the last position you held. 12 to reach the temperature of the room. A day for the bacteria you fed your whole life to stop being held back. 5 days to inflate and then to collapse. A month to turn the ground green. A year to bear the bone. A century for the bone itself to go. None of this is theory. Every stage has been measured on real bodies, by people who donated themselves so it could be. You do not get to opt out of any of it. The only variable you control is where you are lying when it starts.